Hello, today I'm going to be discussing the CRISPR-Cas9 for cancer research and therapy. So within this article, I found a lot of interesting facts on how this specific research will enhance further cancer researches along the line, which I thought was really interesting and cool. So the article first starts talking about how important it is for making changes to genomes of many organisms. And that can be on page 1. It was first discovered in bacteria, and it functions to activate or repress the expression of genes. And then it dissects mechanisms of tumorigenesis, identifies targets for drug development, and possibly arms cells for cell-based therapies. So in the introduction, it talks about how cancer is one of the main deadly diseases that are associated in this world that we live in. And a main pillar of innovation in cancer therapy has been improved understanding of the underlying tumor biology. And this knowledge has led to the development of small molecules and antibodies targeting key proteins of oncogenic signaling pathways. So while these agents have improved survival for cancer entities, for many other treatment options are still limited, and the resistance of mechanisms are poorly misunderstood. So there's just a large scale of sequencing projects that have revealed genetic alterations that are specific to a certain type of cancer or many cancers. So the systematic functional analysis of genes and mutations have been very slow and laborious, which recently the genome engineering was greatly accelerated by the development of CRISPR-Cas9 technologies. This CRISPR-Cas9 can be used to explore the non-coding genome of cancer. So they go furthermore into discussion how in the novel they describe it as in vitro and in vivo cancer models that can be engineered by CRISPR-Cas9. And if you flip on to the next page, it's the first topic, which is CRISPR-Cas9, a versatile tool for genome engineering. So, and two distinct RNAs that demonstrate these studies are CRISPR targeting and trans-activating RNA. There are also cRNA and tracrACRNA, which activates and guides Cas proteins to bind to viral DNA sequences, which are subsequently cleaved. So combining these cRNAs, which are the CRISPR targeting RNAs, with the tracrRNAs, which are the trans-activating RNAs, into a single guide of RNA, further simplified the system. So in 2013, they made a study with Streptococcus pyogenes, and this was used for RNA and DNA cleavage in mammalian cells. And it goes on to explaining before cleaving the target DNA, the Cas9 nuclease undergoes conformational changes in the sgRNA binding, and it's regulated to its target site. So after unwinding all that DNA, the binding to the PAM and DNA hybrid formalization, two nuclease domains introduce two double-strand breaks in the target sequence. So in a contrast to all that information, the homology-directed repair, which is the HDR, uses assisted recombination of DNA donor templates to reconstruct cleaved DNA. This mechanism can be exploited to introduce well-defined mutations by altering donor templates into the targeted cells. So the systemic modifications of sgRNA scaffold identified optimized scaffold structures associated with a higher binding efficiency of Cas9 to the target DNA. So in addition to all of this, CRISPR interference and CRISPR activation exploit fused transcriptional regulators to repress or induce a transcription when dCas9 is directed into the transcription. In most cases, the CRISPR interference systems rely on Cas9 fusions with a Crab repressor domain to downregulate the gene transcription. In contrast to this, the CRISPR activation systems have been using diverse strategies to enhance gene expression. So to investigate the epigenome, the Cas9 fusion proteins have developed that allow the region-specific epigenetic modifications. So furthermore, going into that, recently the Cas9 variants have been engineered that introduces specific single nucleotide base changes. These base editors do not introduce DSBs, but they rely on Cas9 nicknases coupled to cytidine deaminase domains to induce targeted transitions from C to T and G to A. And then it goes on to an example which Komor et al. Designed an efficient base editor by fusing Cas9 nicknase to RAT ABOBEC1, an inhibitor of a base excision repair. Although this approach was limited by a smaller number of potential in sgRNA, it offers the alternate solution to avoid viability, which is repetitive DSBs when targeting amplified genes. So under the next topic, the design of sgRNAs and bioinformatics resources, it starts talking about how they use Cas9 as a rapidly informed tool that keeps on increasing in knowledge for many experiments. The algorithms that determine the sgRNA on the target efficiency are derived from large screening data and integrate both the nucleotide composition of the binding site as well as the relative position within the gene model. In contrast, predicting the specificity of the sgRNAs is more difficult than as most off-targets do not detectable in a detectable phenotype and thus need to be identified experimentally. But the most important thing that I want to take out of this section is the most important lesson learned from these experiments is that off-targets share sequences similarities with target site, but precise bioinformatic prediction of all off-targets is not possible based on currently available data. And then if you flip the page to page 108, Figure 1 expresses in sections of A, B, and C. A expresses the Cas9 nucleases directed to the target DNA by complementary pairing with its bound sgRNA. B, the structure of sgRNA. It's a structure of sgRNA. And C, the selected SP Cas9 fusion proteins with distinct biological effects. So the DCAS fusion to the VP64, transcriptional activator to green to activate the transcription of target genes by binding upstream of transcription start title. DCAS fusion Crab repressor red to downregulate transcription by binding the transcription start site. And Cas9 fusion to epigenetic modifiers. So basically that's what I went over in the first section on page 107 under CRISPR Cas9, a versatile tool for genome engineering. So number 4, the target discovery by CRISPR Cas9 screens. So these are very powerful functional genomic tools to discover novel targets for cancer therapy. And this involves several bioinformatics environmental steps. So the first step is the sgRNAs are predicted to be efficient and selected for every target gene. And then the sgRNA library is synthesized as a pool of oligonucleotides and cloned into lentiviral plasmid backbone followed by a generation of viral particles. These lentiviral particles are then, going on to page 109, are used to infect Cas9 expressing cells at a low multiplicity of infections so that every cell potentially carries out a sgRNA cascade and specific gene knockout. Subsequently, this pool of knockout cells is kept in culture for a fixed period of time or exposed to selected perturbations, which are harvested and their genomic DNA is extracted. So within that, one main goal of pooled CRISPR-Cas9 screens in cancer research is to identify the genotype-specific vulnerabilities. These essential genes can be potential drug targets as their functional depletion leads to a reduced viability. And going on furthermore, on that same page 109, figure 2, it shows the pipeline for generation of knockout cell pools. So this is an overview of the individual steps in the design and the cloning of sgRNA and sgRNA libraries and generations of knockout cell pools. So these appropriate sgRNA binding sites are identified in silico by library design software tools. So as you can see, section part A, a pool of oligos containing the sgRNA are produced by the chip-based synthesis. B, and cloned into a lentiviral backbone vector. C, a lentiviral particles, each containing a distinct sgRNA expression. And D, after antibiotic selection, the pool contains cells with distinct genes knockouts, which can be used for a variety of different approaches of screening. Then going on to the third paragraph of page 109, they express that the concept of gene essentiality can be narrowed down to gene regions encoding the essential protein domains. As opposed to RNAi that depletes the transcripts of target genes, it is possible to disrupt the specific protein domains using CRISPR-Cas9 by targeting the corresponding genomic sequence. So then from there they use the process called the screening approach. So this screening approach is termed domain screening, was used to pinpoint protein domains that determine the essentiality of a gene, which can further facilitate drug development. A complementary screening approach to identify druggable targets relies on the concept of synthetic lethality, which is a specific form of lethality that occurs if a cell can compensate the function of alteration A, gene A or B, but is unable to survive and gene A and B are simultaneously altered. This means that previously non-essential genes can become essential due to the loss of a specific tumor suppressor or an activation of an oncogene. So going further into that, they did a clinical trial that showed the higher sensitivity of breast cancer bearing a germline mutation, BRCA, and PARP inhibition. Several CRISPR-Cas9 screens were performed to identify the synthetic lethal interactions. And then but more in similarity, they used a similar approach which was used to identify the targets in leukemia cells that harbor the MLL8F4 fusion oncogene by means of genome scale CRISPR screen. The previously uncharacterized gene ENL was found to required for growth and proliferation in MLL8F4 positive AML lines. Flipping the page to page 110, this furthermore goes into in figure 4. It shows the concept of essentiality, and it just goes over the overview of the different mechanisms of essentiality in cancer. And in figure 3, it shows the pooled CRISPR-Cas9 screening approaches, and there's 3 different screening approaches which are presented in the form of a diagram. So A is the pool of knockout cells is incubated for defined time span. B is the pool of knockout cells is separated and treated with a compound or a carrier solution for a defined time period. And C, the additional highly expressed barcode is encoded on the lentiviral vector. So that's just an overview of those that I went over. And then turning on to page 111, going into section interrogation of the non-coding genome of cancer. This paragraph describes the first paragraph describes how like the mass majority of human genomes consist of non-coding regions that contain many regulatory elements, such as enhancers or non-coding RNAs. And specifically in cancer, the expression of non-coding RNAs is known to be dysregulated, and the transcription of oncogenes can be controlled by near or distant enhancer elements. So, to identify these enhancer regions of 3 cancer as associate genes is CUL3, NF1, and NF2. So a CRISPR screen was performed to targeting genomic regions of 700 kb surrounding the open reading frame of these candidates. In fact, the alterations of specific genome sites down and upstream the CUL3 gene changed occupancy of the specific transcription factors and reduced CUL3 expression. In a similar approach to this process, CRISPR-A was used to identify LNC RNAs that mediate resistance to BRAF inhibition. So several candidate loci were found within that, and the result reflects one of the major caveats of the CRISPR-I/A to target in the LINRNAs, since many LNC RNAs have bidirectional promoters that are located within the protein coding genes. Then going on to under the section of genome editing to generate the organoid cancer models, it explains how the adult stem cells derived from organoids are increasingly popular in the in vitro model of the intact and diseased human epithelia. And it just shows how like the possibility to culture both healthy and cancerous tissue enables the study of tumor development in vitro. And then underneath section 7, which is the methods for in vivo delivery, it expresses how the in vivo is an efficient delivery for both of the Cas9 and the sgRNA to the target cell because it's required. Ideally, the method should provide a high editing efficiency and cause low immunogenicity and enable the Cas9 sgRNA to be directed to the organ or cell type of choice. Now, going on to the next page on 112, figure 5 shows the modeling of adenoma carcinoma sequence in the mouse colon organoids. So this is, so A goes on to show the sequential loss of the tumor suppressors and the activation of oncogenes lead to enlarged, highly proliferative organoids with an enhanced capacity to form colonies. And B shows hyperactivated WNT signaling by loss of APC leads to independence from WNT activating growth factors in the medium. And furthermore, going into that, the adeno-associated virus, the AAV, is recorded as a potent tool as they are non-integrating, possess a high transduction efficiency, and are serologically compatible with a large fraction of the human population. These AVVs is characterized by rich diversity of serotypes with distinct tissue tropisms enabling the selective targeting of different organs. Similarly, direct injections of ABVs into the striatum of mice can locally modify the Huntington gene. And this approach has been successfully used for editing the ornithine transcarbamylase gene in the murine liver and the dystrophin gene in the muscle tissue. After systemic application, the editing frequency ranged from 10% to 70%, which was sufficient in the phenotypical improvements of the mouse of hereditary diseases. Going on further into that, an alternate approach to the viral delivery is the application of the lipid nanoparticles. A major advantage of the nanoparticles is that it can be manufactured on the industrial scale, allowing a dosing that is more similar to commonly used drugs. Flipping the page into page 113, figure 6 gives a great example of the pooled CRISPR screens in the mouse, and it's illustrated by Shen et al. Non-metabolizing Cas9 expressing cancer cells were infected with genome-wide sgRNA library ex vivo. And then that just gives a basic understanding of what goes on during that time. And then going on to section 8 of in vivo, instead of in vitro, it's in vivo genome editing and genetic screens. There's two basic concepts, and these two basic concepts are the implanting of the ex vivo edited cancer cells, and the second one is the in vivo delivery of CRISPR-Cas9 components to mouse tissues. They transduce a non-metastatic lung cancer line with an sgRNA library and subcutaneously transplanted these cells into the mice. The implanted cells formed the local tumors and lung metastases, which were extracted and analyzed for the enrichment of specific sgRNAs. So in this direct in vivo genome editing is a potent approach to studying the effects of genetic alterations of carcinogenesis in a native tissue of a microenvironment. The first evidence that in vivo gene can result in tumor formation was provided by XUE. I'm not sure how to pronounce that. And in that study, the plasmid encoding Cas9 and sgRNAs against P10 and TP53 was transfected into murine liver cells by hydrodynamic tail vein injection, resulting in the formation of liver cancer. So as you can see, all of these things start adding up and are shown within these experiments. And going furthermore onto the next section, section 9, the first clinical applications of CRISPR, it shows that the new technologies of the gene editing may have overcome some current issues and allow permanent modifications of somatic cells. As studies in animal tissues, both in vivo and ex vivo approaches are studied. So this is the first clinical trial showing the feasibility of the gene editing in the clinical setting was published in 2014. And they did not use CRISPR, but they used zinc finger nucleases, so ZFN. In this trial, the major HIV co-receptor CCR5 was targeted by ZFN in autologous T cells ex vivo to induce the resistance to the infection with the virus. The study then revealed that the tolerability of the CCR5 modeled cell infusions, and indeed CCR5 knockout cells declined significantly less upon the viremella than the unmodified counterparts. So the first clinical trial using CRISPR for cancer therapy has been enrolled in the first patient in 2016 at Shuaishan University's West China Hospital. And in this non-randomized open-label phase 1 study, this safety of the program's cell death protein, PD-1, knockout engineered T cells ex vivo is evaluated in treating metastatic and non-small cell lung cancer that has progressed after a standard treatments. So even though this concept is promising, there are many doubts with PD-1 knockout engineering T cells are preferable over PD-1 or PD-L1 antibodies in a clinical setting, since the processes of genetically modifying, propaganding, and infusing T cells is laborious. Therefore, under section 10, this talks about the future of the CRISPR for cancer research, and it just basically goes on to say how this can be something that we can use to treat cancer patients, as it is a very informational access tool for biomedical research. And right now it is we expect to see unexpected results. We don't see exactly all the results that we would like within this research, but going furthermore into it, it is definitely something that is ongoing. And if we, as it says in page 114, I believe, if we can understand the biological mechanism underlying the many genetic interactions revealed by CRISPR-Cas9 screens, this will provide a tool to manipulate non-coding regions of the genome and will accelerate the functional exploration of a so far poorly characterized aspect of the cancer genome. So finally, we expect to see the yet unpredictable results of the pilot clinical trials and as a therapeutic tool to target the immune system against cancer. The future use of CRISPR-Cas9 in translational medicine will largely depend on the ability to develop the Cas9 variants with minimal or no off-target effect and novel methods to improve yet the efficient engineering or the precise genetic changes by the homology-directed repair. So this is a very important, I believe that it's a very important article to come across by, because cancer is something that is so devastating, and it's one of the most deadly diseases that we have in this world. And if we have this technology, we should be able to furthermore go on to information about it and be able to advance it and hopefully find a lot of cures for cancers and fully understand how cancers work and how we can prevent cancers. So I think that within this article with cancer research and therapy, this is definitely CRISPR-Cas9 is something that we should furthermore look into and make sure that we don't stop because we don't get answers, but keep on going with this process to see if this CRISPR-Cas9 could really, really help us with cancer research. That is it. Oh my gosh, 28